All right, today we are going to do a visual deep dive into the adaptive immune system. We're going to map out the entire process, step by step, so you can actually see how your body's most sophisticated defense force gets the job done. Let's get right into it. So, here's our game plan. We'll start by meeting the special forces of the immune system. Then we'll see how they're trained, how they get the alert when there's a threat, how they coordinate the attack, and finally, and this is the really cool part, how they build a lasting defense so the same enemy can't hurt you twice.
Okay, Section 1, the body's special forces. So you can think of your body as having three main lines of defense. The first two, well, they're part of your innate immunity. They're super fast, but they're kind of generic. We're focusing on that third line of defense, the adaptive immune system. This is the highly specialized elite force.
And this slide really nails the key differences. Your innate immunity is like the first responder on the scene. It's immediate, but it treats every problem the same, and it doesn't remember anything. The adaptive system, though, is different. It's inducible, which just means it has to be woken up and activated, so it's a bit slower to start, but its response is incredibly specific to each threat, and this is crucial, it remembers every single enemy it ever fights.
So who are the key players in this elite squad? They're the specialized cells you see right here, the lymphocytes, shown in yellow. They, and the amazing proteins they create called antibodies, have one single mission: hunt for very specific targets called antigens. You can think of an antigen as a unique molecular fingerprint that's found on an invader, like a specific part of a virus or a bacterium.
On to Section 2, training the army. Now, before this elite force can actually go out and fight, it has to be built and it has to be trained. This is where we forge the two main types of immune soldiers, the B cells and the T cells.
This diagram gives us a fantastic bird's-eye view of the whole process. It all starts with precursor cells that are born in the bone marrow. Some of them stay right there in the marrow to become B cells, while others travel to a special organ called the thymus to become T cells. These are the primary training grounds, kind of like boot camp. Once they're trained, they get deployed to secondary locations like your lymph nodes, where they basically just wait for the call to action.
So first, let's zoom in on B-cell training in the bone marrow. It all starts by creating a massive amount of variety. That's called clonal diversity. Basically, each B-cell gets a totally unique receptor. But here's the most critical step of all, the self-antigen test. Any B-cell that reacts too strongly to our own body's tissues is destroyed. This process, called clonal deletion, is how we establish tolerance and prevent our immune system from attacking us. The survivors are now fully trained, but they're considered naive, just waiting for their very first mission.
Now, T cells go through a very similar process, but their boot camp is in a different place, the thymus. They also generate that clonal diversity and go through clonal deletion to make sure they don't attack self. The key difference here is that they mature into two distinct types. You've got your CD4+ helper T cells, who are like the coordinators or generals, and your CD8+ cytotoxic T cells, who are the direct hand-to-hand killers.
And just to really drive home how rigorous and strict this training is, get this. Over 90% of all developing lymphocytes are eliminated because they are self-reactive. The body's quality control is unbelievably strict, all to prevent friendly fire.
Okay, Section 3, sounding the alarm. So our army is trained, it's deployed, and it's waiting. What happens when an invader finally shows up? Well, the first step isn't to just attack blindly. It's to sound the alarm and show the troops exactly who the enemy is. This critical step is called antigen presentation.
To really visualize this, picture a scout at the site of an infection. That's this dendritic cell. It's a professional antigen-presenting cell, or APC. Its job is to engulf a pathogen, then physically travel through the lymphatic system to the nearest command center, a lymph node, where it can present the evidence of this invasion to all those naive T cells that are waiting there.
So what's going on inside that APC? Well, the pathogen it swallowed gets broken down into little tiny fragments. The APC then takes one of those fragments, the antigen, and loads it onto a special display platform on its surface called a major histocompatibility complex, or MHC molecule for short. Now, there are two types of these display platforms, and they signal very different kinds of trouble. MHC class 1 is found on almost every cell in your body. It presents internal antigens. You can think of it as a cell raising a flag that screams, "Help, I'm infected from the inside, please kill me." This is the signal that activates our CD8+ killer T cells. MHC class 2, on the other hand, is only on those professional APCs. It displays external antigens from pathogens they've captured outside. This is like the scout holding up the enemy's uniform and saying, "This is who we're fighting." And this is what activates our CD4+ helper T cells.
And that brings us to the command center. The T helper cell is the master coordinator, the quarterback of the entire operation. It doesn't kill anything itself. Its job is to assess the threat presented by that APC and then orchestrate the entire immune response.
Activating this general isn't simple, and that's a really important safety feature. First, the T helper cell's receptor has to bind to that antigen MHC complex. But that's not enough. It needs a second co-stimulatory signal from the APC. It's like an "are you absolutely sure" handshake. Finally, chemical messengers called cytokines are exchanged. Only after all these steps have been completed does the T helper cell fully activate and start to multiply, building up its numbers.
Once it's activated, the T helper cell differentiates into specialized subtypes, kind of like a general deciding on the best strategy for the battle. For our purposes, the two most important subtypes are Th1 cells, which orchestrate the attack on our own infected cells, and Th2 cells, which mobilize the B cells to start pumping out antibodies.
So, Section 5, the two-front attack. The orders have been given and the adaptive system launches its assault. This is where we see the two main branches of the attack spring into action: humoral immunity and cell-mediated immunity.
Here's a really simple way to remember the difference. Humoral immunity, which is directed by those Th2 cells, involves B cells making antibodies. It's like an air force launching missiles to take out enemies that are floating around in the body's fluids. Cell-mediated immunity, directed by Th1 cells, involves the cytotoxic T cells directly killing our own infected cells. This is the infantry, engaging in close quarters, hand-to-hand combat.
Let's take a closer look at that humoral response. So a B cell recognizes the pathogen with its unique receptor, but to get fully activated, it needs a confirmation signal from an already activated Th2 helper cell. Once it gets that final green light, the B cell transforms into something called a plasma cell, which is basically just a dedicated antibody factory, churning out thousands and thousands of antibodies every single second.
And what do all these antibody missiles actually do? Well, they're incredibly versatile. They can neutralize a virus by physically blocking it from entering our cells. They can activate a protein cascade called complement that literally punches holes in bacteria. And they can act as little tags, a process called opsonization, marking invaders for destruction by other immune cells.
And finally, after the battle is won and the infection is cleared, the most remarkable feature of the adaptive immune system comes into play: its ability to remember.
The battle may be over, but the war is never forgotten. The system doesn't just reset back to zero. Instead, it records the entire experience, creating a powerful, long-term, and highly specific defense.
Here's how it works. A fraction of the activated B and T cells that fought so hard during the infection, well, they don't die off. They become long-lived memory cells. These are the veteran soldiers, and they circulate in your body for years, sometimes for your entire life. If you are ever exposed to that exact same pathogen again, these memory cells are ready. They launch a secondary response that is so fast and so overwhelming that the invader is wiped out before you even realize you're sick.
And that, right there, is the whole scientific principle behind vaccination. We introduce a safe version of an antigen to build this army of memory cells without ever having to get sick in the first place. This powerful, specific, memory-building system is truly one of biology's most elegant designs. And it leaves us with a really exciting question. How else can we harness it? What's the next frontier for training our bodies to fight things like cancer, or for teaching the system to calm down in cases of autoimmunity? That is where the science is headed right now.